So Pankaj Agarwal, he's a senior fellow at GSK. He's one of our advisors. He's a computer scientist, computational biologist. So he will talk about predicting therapeutic targets from pathways and omics data. So thank you for coming again for many years now.
That's right.
We're glad to have you.
Thanks, Stefan. It's great to be here and see all the science that's happening. So let me, okay, so let me start by reminding you that developing drugs is really hard, right? People talk about making Hollywood blockbusters being hard, but they got nothing on developing drugs. The success rate of taking something that's in the clinic, is in a phase 1 trial, and making it to market is only about 10%, right? And that's, so that's both in some ways shows you how challenging the problem is. That's why we want as many people to work on this as possible so we can find more medicines. And at the same time, it also tells you that, you know, when people find great results, when you see a model that says I have a 98% accuracy of predicting or 94% accuracy of predicting what target will make it into a drug, you know, you should think about that a little bit, right? Because clearly that's a little bit impractical. So what we are looking for in this state is just, you know, a little bit of improvement here can make a huge difference, and I'll show you as to what that is and how we get to that. So for drug discovery, the way I think about drug discovery, there are 3 basic entities I deal with. Those are patients or diseases, right? And what proteins or targets are involved with them? What targets should we develop drugs around so that we can help those patients or those symptoms for that disease, right? So that's one question. The other question we have is clearly there are drugs that target these proteins, right? So this is a very simplistic model of the world. And so if you have these drugs that target these proteins, and these drugs can be used to answer many questions about these populations. So you can find what population is best affected by the drug, called drug repurposing, the first indication. You can also find additional indications for that drug, what other patient groups that can be helped by that, and that's a drug repurposing problem. And you know, people mention these problems, and we have seen some data on this at this conference as well. Also, population enrichment. You don't want to necessarily do a trial where you treat the largest group of people for the first time and see whether your drug works or not. That's very expensive, right? What you want is a smaller group of people where you can show that the drug has a better chance of success and you can get a stronger signal, right? You want to make sure that it's powered correctly. And you want to have biomarkers, right? So you want to be able to say which patients I should test this drug in, and that's where omics and a lot of these technologies come in. If you only need to test people who have a BRAF mutation for that drug, it clearly simplifies your life. On the other hand, you also want to know who's going to respond to it. So trials can be very long, right? So you want to be able to see, is this drug likely to work in this patient? If it's not, let's get them off this drug and get them onto something else that might work for them, right? So this is a problem around biomarkers. And these are all problems that can be solved with the data that you guys are generating and analyzing, right? So the links data, I think, plays a huge role in many, many of these problems. In addition, from the drug, you can also figure out what's the target of that drug. So a drug may not necessarily just bind to one protein, it might bind to others. Mechanism of action and the links, the L1000 data especially, even the imaging data, you can see the applications of that to figuring out what the additional targets might be there. And of course, then when you have these targets, you want to screen these to find these drugs in the first place, but that's typically not a computational biology problem, so we'll skip that for today. Within populations, understanding heterogeneity, what's the right subpopulation, what exactly is a disease, even that's not very clear, right? So diseases have been labeled by physicians based on symptoms for a long, long time, but in terms of treatment, is that really the best approach? Or molecular markers or imaging markers, those are better, right? So how do we define those groups? I think electronic health records are playing a big part in that, and again, we would love to see more and more work around this area. But for today, what I'll talk about is mostly around target identification, and I'll tell you in a few minutes why. But before I do that, I want to give you a little vignette on this genetics-based repurposing that we did a number of years ago, and you'll see later on how this connects up. So there was a fantastic talk yesterday around why GWAS studies have not lived up to the promise and hype, right? And you know, that's a view that's widely shared. The odds ratios aren't fantastic. We've invested all this effort. What have they given us, right? So we did this study a few years ago where we looked at genome-wide association studies, all the published genome-wide association studies at that stage, and we intersected them with the industry pipeline as to across all pharma and biotech, what was the industry pipeline, and said what proteins are in common. Which are the proteins or genes for which we both have a association to some trait, and we also have actually a drug in the pipeline for any of the companies. And we found there were 155 genes at that stage that actually shared this overlap, that had were both the targets of a drug and had an association study. And then we asked whether this, and that was actually pretty interesting because it showed that, and this was more than you expect by chance, which showed that, genetics and pharmacology do seem to intersect, right? But then we asked the question, which of these traits that actually matched the current indication for the drug, right? Does the genetics suggest the same disease or a different disease than the drug does, right? And we found that for 63 of them, it was the same indication or a related enough indication that you could call it same. And, that's good. So clearly that the drug was developed for something that the genetics was supporting. It doesn't really matter what came first. And for another 92, it was potentially a new indication that based on genetics that you could test out, right? So you got a lot of repurposing opportunities out of that. But if you think about that a little bit carefully, that's something that bothered me then and still bothers me, is that, if you look at where the match is, that's exactly what you would expect. So this is 2.5 times. 63 is exactly 1 time of what was expected, and this 92 is the surplus 1.5, right? So if you actually match the indication, genetics did not actually predict the indication. All it said was genetics-based targets are good targets for either that indication or some other indication, right? Which is not exactly what you might want, right? But we'll come back to this. So let's go back to targets. Why are we interested in finding targets, right? Majority of phase 2 and phase 3 clinical trials fail due to lack of efficacy, right? Because we cannot show that the drug actually has enough of a response in the population you studied in, right? The other thing to remember with all this stuff is failure of trial doesn't mean failure that the hypothesis was wrong, because these are all one-sided hypothesis checks, right? And most of these were likely due to choice of poor target, because hopefully it was powered enough and you had enough target engagement. So if your phase 2 or phase 3 trial is failing because of lack of efficacy, it's often due to poor choice of target. I mean, that's an assumption that we could question, but it's widely believed that's the case. So what can we do about this, right? So a few years ago, there was a paper from some of my colleagues at GSK, and AstraZeneca also published a similar paper with very similar results, showing that genetics-based targets actually had a higher success rate in the clinic, right? So if you look at targets that had genetic support, you could get 2-fold higher success rate from phase 1 to approval, right? So that gave us a clue that maybe genetics does provide the kind of evidence you need that can lead to more successful drugs, right? So this was based on successes versus failures. So we also wanted to go a step further and look at what does that really mean. So say you had genetics-based targets, or you had some other technology that gave you 2-fold higher success rate. What does that actually mean in real terms, right? Does that really change anything in terms of medicines? So we did a small study where we actually looked at what the impact of these targets would be if you instead of having, say, 15% genetic targets in your portfolio, your entire portfolio was these better quality targets. And we showed that you can get actually 38% more medicines. So even without spending any more money, if you just picked your targets better, you can get 38% more medicines out of the gate. And of course, that takes 10 years or so to get them out of the gate, but you could actually do that. So improving targets either through genetics or other means, has a real impact on medicines, right? But let's get back to genetics. So then how do we do this, right? So you can pick genetics-based targets, and they'll have a huge impact on the number of medicines. But most genetic targets or GWAS genes are not druggable, right? You cannot easily make small molecules that bind to them. The other bigger challenge that almost nobody talks about, the GWAS gene itself doesn't give you a direction as to what you want to do. You don't know whether you want to upregulate that gene, downregulate the gene, whether it's because the function of most of those mutations is not known, right? So it does not give you pharmacological direction, right? And especially in the cases where it's, say, downregulated, it's a loss of function. To replace the function by a drug is pretty hard, right? Other than through means like gene therapy or others. So the idea we had, and this is not a novel idea, that why not target something else in the pathway, right? Why target the GWAS gene itself? Of course, the challenge is you can go on it a case-by-case basis and say, look, here is a GWAS gene. I know this is what upregulates it. We can make a drug against that, and it might work. But can you show it systematically that something like this might actually have value? So what we did was we took one of the more comprehensive collections of GWAS data called stopgap. And we filtered it for the highest quality associations that were p-value 5e minus 8. We looked for traits that actually had 5 or more genes or loci associated with them. And what we did was we also looked at a resource for pathways, and we used Metabase, which is a commercial resource, but you could use something else as well, which had about 6,500 genes and about 1,500 pathways, because it was the largest collection of pathways we could find that was quite comprehensive. And then for each trait and their genes, we did a pathway enrichment, right? So we did 278 pathway enrichments across each of these traits, saying what pathways are picked up for each of these traits, right? And we did FDR and all that to make sure we weren't overcounting. And what we found was that 202 traits for which we had 1,165 pathways, right? Clearly some pathways showed up for more than one trait, but for many of these traits, you could find pathways that represented the genes that were part of the GWAS loci. One other thing I should tell you is when we did the GWAS gene analysis, we did not just take the top gene for each loci. We took all the genes in each loci, and we let the pathway actually find what the correct gene might be, or what sort of was the most generous interpretation of the GWAS data. And then we corrected to make sure we didn't overcount those numbers, right? And we can go over the details later. So what we found was there were 182 diseases of those 202 traits. There were 20 non-disease traits. There were 182 diseases that led to these enriched pathways. And we think that this can lead to new genetics-based biology, right? So if you look at those pathways, now you have a better understanding of what the GWAS signal is actually pointing to. But so what? I mean, you can claim this, and does this really make sense, right? So how do you know that what you have is real? So what we did was we did a in silico validation where we took these 182 diseases with enriched pathways, and we intersected this with the known drug targets, right? So Metabase resource also provides known drug targets, and we found that there were 538 diseases which had drug targets, and when we overlapped them, we found that 97 of these 182 actually had known drug targets from Metabase, right? So these diseases now had pathways and had known drug targets, and 85 had no known drug targets. So let's think about these 97 diseases with drug targets. We asked the question, are those, so you start with a disease, you look at its pathways, and you look at the drug targets in those pathways. Are those drug targets in those pathways indicated for the same indication? Remember our earlier problem that we could, even though we found genetic enrichment, we could not show that they were indicated for the same indication. And we found that for 29 of those diseases, that you actually could show that was indeed the case, right? And remember in the GWAS-based example, that number was 0, right? So if you actually FDR correct for it, if you just do GWAS-based alone, you do not see any enrichment. So this is the GWAS-based data, right? The green is the total number of drug targets in each of these diseases. The blue is what is discovered by GWAS alone. And if you add pathways, the blue suddenly rises. And for 29 of these diseases on the x-axis, the blue is a significant fraction of that. So we think that applying the pathway approach can actually lead to enrichment for potential drug targets for that disease, right? Of course, not all of these are going to be real, right? So there's still a lot of work to do. So just to walk you through an example, Behçet's is a inflammatory disorder. It's reasonably rare, but there's a GWAS published on it. There are 19, there are 19 loci linked to Behçet's disease, and if you do a pathway enrichment, we found 12 enriched pathways, right? And in these 12 enriched pathways, there's 7 drug targets listed for Behçet's in Metabase, of which 6 we could find using this approach, right? But these pathways, these 12 pathways actually have a lot of other targets for a lot of other drugs. In fact, one of them is a target, is a drug for IL-12B, and there's a marketed drug for it. And when we looked at this further, we found that actually somebody was already doing an open-label study of that drug for Behçet's disease that was not captured by Metabase, right? So you can certainly look at some of these other things, and they do make sense, right? Not to say that everything in there is going to make sense, but there's some good signal in there. All right, so so genetics can, hopefully I've shown you some data that genetics or GWAS-based studies can lead to good drug targets. But, why just genetics? I mean, you guys generate tons of omics data. We all analyze tons of omics data. What makes genetics so special, right? So we asked the question, can we find the same kind of signal for other omics data, right? And this is something that Andrew Roulard, who actually came from Avi's lab, did, and he's now at GSK. In fact, I met him here 2 years ago. So So something concrete does come out of these meetings in addition to all the great science, right? So what he did was, I mean, as you know, Andrew had worked on harmonisome at Mount Sinai. So we took the data from harmonisome that GSK had legal access to. So there is another problem. When you integrate data, many of these things are semi-commercial resources that we cannot just bring in and use. We have to make sure every resource within them we actually have access to. So you think you're creating a fantastic resource, but it's still sometimes a little bit painful for us to figure out what we can use and what we can't use. So he took that data, right, and that has data around all the targets. And Mark Hurl in our team actually had collected a really nice set of 331 phase 3 clinical successes and failures, where we, for 72 of them, they had failed in phase 3, and for the other, whatever the number is, they were actually successful in phase 3. And this is sort of the targets that we had, the sort of the cleanest data around phase 3 success and failure. And it's sort of your gold standard data set in terms of phase 3 success and failure. And pretty much you can use this for any classification problem. Now, can you predict success and failure in phase 3 by combining it with any data set that you would like to see, right? So, and this data set is already on bioRxiv and GitHub, so anyone can have a go at it, and please do have a go at it, right? So what he did was, just to show you, is, and this is tricky to clean up this data, right? So, I mean, just to be, it's not that easy to assemble. But what he did was he took those 331 targets and collected 28,000 features from harmonisome and said, would any of these features predict the phase 3 clinical success and failure, right? So you can do a fairly standard feature selection pipeline, and I'm sure you guys can come up with other ones. You can. What we were looking for is significant, robust, generalizable features. You know, I started with a slide on the success. The success rate is so low, and most of these models predict fantastic results. We wanted to be very stringent as to what we predicted. So we used very serious cross-validation. We used really robust understanding of what these features were that would show up, and we disqualified anything that did not hold up according to at least 3 different cross-validation criteria. And all that's in the bioRxiv paper if people want to have a look at it. And we also actually took those features and tried to build a model around them. Again, the emphasis here was on building simple models. So we used logistic regression and random forest, and we did a double-nested cross-validation to make sure that, you know, this was as kosher as it could be. And if somebody spots anything that we can do to make it more kosher, we would love to hear from you, right? So we looked across these 1,000 cross-validation data sets or results to see what features showed up most often, right? So remember, we started out with 28,000 features, and this is the features that got picked, right? So of the 1,000 cross-validation folds, the mean expression across tissues was picked up 685 times, right? And the standard deviation across tissues was picked up 585 times, right? And so remember, these are not the features we have came directly from harmonisome. We did not sort of hand or custom adjust them, right? Clearly, you could custom build or custom pull different features from different sources and get other things to show up. This does not mean that other features are not useful, right? Right? But there are 2 features that showed up time and time again. And when we looked at them, what we see is that if you look at the mean mRNA expression on the x-axis and the standard mRNA expression on the y-axis, that things that have a high mean expression and a low standard deviation, what does that really mean? It means it's expressed pretty much everywhere, are typically fail, right? And things that have a low mean expression and high standard deviation, so probably expressed in very limited things, succeed, right? I mean, this is not surprising, but at the same time, now the data is actually telling you this as well, right? And we had shown earlier that before Vinod had looked at this little study I'm showing, people had actually not even gone back and seen our genes actually expressed in the tissue of interest for that disease, right? Of course, everyone on their project looks at it, right? But systematically, people hadn't checked, is the gene actually expressed? And we found that 87% of the genes are actually at least expressed in the tissue of interest. And now we can say that, look, you clearly want your target to be expressed in a right cell type, right tissue, and you want it to have low mean and high standard deviation. And that might help just a little bit, right? We're not going to go to 90% success rates, but we might increase our, and this is what our cross-validation and our AU rocks look like, right? Or ROCs look like. So it's barely significant, right? You know, most of you would have trouble publishing this data, and we might as well, right? Right. So Uh, so, but we think there is enough of a signal here that there is something in there, and moreover, it seems to make some sense, right? So, uh, can I just take the questions at the end? And I'm almost at the end. So just to wrap up, so hopefully I laid out some key questions in drug discovery for you, right? So you can think about those. Those are the things that interest us. The data you guys generate and the tools you guys make, we use them a lot, right? So we are fanatical consumers of the stuff you guys produce, right? So nothing we do is built completely by ourselves. It stands on your shoulders. So hopefully I also showed you how you can find targets from GWAS using pathways, right? And it lays out some elements. I mean, sure, there's a lot of work to be done there, but again, we look forward to people trying it out at other different pathway resources, seeing what works, what doesn't work, and evaluating successful target properties. I just want people to really be beware of models with very high success rates, right? So when I see a repositioning paper and they claim they got 92% accuracy, one thing to think about is you want a model not with high area under the ROC curve. What you want is a model where when you look at the top 100 predictions, the number of testable predictions is what your next assay can take, right? So if your next assay is an imaging assay that Anne is running, maybe you can do a few hundred tests, and you want to make sure there's enough signal in there that she gets a good result. Or if the next assay is actually a human clinical trial, maybe there are 5 hypotheses that can be checked, right? And that's being generous. Make sure there is 2 good ones in those 5. So you want to have your precision for the early part really be high, right? Or not. When I say really high, I just mean 20%. And please use other data and techniques. The data set is out there, both the gold standard success failure targets as well as the harmonism. Of course, there are better ones. And of course, the other part is don't stop at the hypothesis. We have all these resources these days. From disease, you can use Pharos, open targets, many of these platforms to find targets. From targets, there are multiple resources to find tools that you can use to interrogate them. There are lots of resources to find models, right? What's a good model for a particular disease? Actually, this is probably the weakest area of this whole, is that the good disease-relevant models are very tricky, right? And there are very few I'm aware of, and if somebody has good disease-relevant models that translate into humans, certainly want to hear more about that. And then you can use those models to test these tools and verify your hypothesis. So don't just stop at generating your hypothesis, actually test them and experimentally validate them. I'm going to stop here and acknowledge my collaborators. Dipali Jham led most of the work on the GWAS pathway analysis. Andrew Roulard, who many of you know, did the harmonisome target success failure thing. Michal and Mark, Bill Reistoff, and Philippe, they were involved with much of this analysis also. I'll stop here and take questions. Thank you. Yeah, no, that's a great question. Looking at specific disease spaces, can you actually find different features in those spaces, right? The challenge is that the number of examples is already so low. We only have 332 or so examples. So if you reduce it by disease space, it gets to be even much fewer. So build a model on each of them. It will require even some more nimble machine learning that maybe some of you are capable of. One thing we did do that I didn't mention was we looked at it by target class. Right, so among the checks we did was, for example, GPCRs have a much higher success rate in phase 3 than do integrins. There were 3 integrins that have been tested in phase 3 that have all failed, and GPCRs have a 90% success rate in phase 3. But is that kind of feature really important in terms of predicting success rates? Because GPCRs are historical targets; they've been used for a long time. Maybe so it's a little bit. It can be quite interesting in terms of how you interpret this data. Yeah. 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 No, that's an excellent point. There is a lack of gold standard in this area, and that's not for a lack of effort. I think what we have is multiple bronze standards, right? So you can have a phase 3 success failure rate. You can do a phase 1-2 approval success failure rate. Also, I left out a lot of details. What do you do with polypharmacology drugs? What do you do with drugs that target multiple indications? How do you handle things that have failed for one and succeeded for another? So there's a lot of different slices and dices you can do of this data, and the results can vary, right? So I suspect we'll never get to a gold standard data set on this. What we'll have is multiple bronze standards and hypotheses or features that make sense, right? And then you will start applying them prospectively and see if they work out. Yeah, so the data set, so this is one area where there's a commercial resources like pharma projects, which are a little bit more extensive in terms of clinical successes and failures, and the tracking is better. As far as we have seen, I think the public resources are certainly getting very good, but they're still not comparable. So we wanted to get sort of the largest set of examples we could get our hands on, and that's why we stuck with pharma projects. But again, so it's worth. Well, what's the target of a drug is a whole different question. We could spend days talking about it, right? So that's a and I think Avi showed or somebody showed that there were differences in all these different databases, and it's hard to know what's real and what's not real, right? So, but the good thing, what I would suggest is try different standards, try different data sets. Essentially, you want to do an all versus all and see what data and what features work for what data sets, right? And because I don't think we'll ever agree on what is the best standard or the best feature necessarily. All right, thank you.